DNA Depot presents How Clean is Our Home? Kit DD301. The objective of this experiment is to determine how effective cleaning products are for homes. You will discover how to safely grow bacteria on agar plates and test cleaning products to determine how effective they are as bacterial growth inhibitors. This experiment contains components to test up to 6 cleansers plus a positive and negative control. Your kit should include bacterial filter paper, a sample of effective disinfectant, Petri plates, sterile bacterial nutrient agar, sterile nutrient broth, sterile swabs, filter paper strips, and tweezers. You will also need bleach to disinfect bacterial plates, various home use detergents, soaps, and cleansers, antibacterial or disinfectant soap for washing hands, your microwave, a large beaker or jar, a hot glove, Scotch tape, a permanent marker, small disposable cups, gloves, and goggles. A. Preparation of bacterial plates. You should do this 1 or 2 days before the experiment. 1. Uncap the bottle of bacterial nutrient agar, also called medium, but leave the loosened cap on the bottle. Loosening the cap will allow steam to vent during heating. 2. Place the bottle in a beaker or glass jar and place it in a microwave oven to melt the medium. Important: use consecutive short periods of heating in the microwave oven to melt the medium. Rapid heating can cause the medium to boil over and out of the bottle, resulting in medium loss. 3. Set the microwave to medium heat and heat for 5 to 10 second intervals. Wearing hot gloves or oven mitts, carefully swirl the bottle in the beaker. Make sure the bottle is loosely capped at all times. 4. Repeat heating for 5 to 10 second intervals and swirling the medium until it is completely melted. This should take approximately 30 seconds. The medium should be a clear yellowish liquid. Remove the medium from the microwave with oven gloves. 5. Let the medium cool at room temperature until the bottle is very warm to the touch but not burning hot. 6. Lay the Petri plates on a flat, clean surface with the lid on top. 7. Lift the lid and pour the melted medium, 5 milliliter each or approximately equally, in the 5 Petri plates provided, so the bottom plate is covered with a layer of medium, at least 1/3 full. (8) Quickly cover the plates and let the medium solidify for 1 hour. You may store plates inverted at room temperature until the day of the experiment. If the plates will not be used in 2 to 3 days, they should be wrapped in plastic wrap and kept in a cool, dry place. B. Preparation of bacterial suspension. This should be done 24 hours before the experiment. A safe Bacillus species bound to filter paper has been provided in a small capped tube. 1. Carefully unscrew the cap of the sterile nutrient broth bottle. 2. Transfer the bacterial filter paper directly into the broth by carefully emptying the tube containing the filter paper. 3. Cap the nutrient broth bottle and invert several times to mix. 4. Place the nutrient broth bottle in a secure place and incubate at room temperature for 24 hours. 5. After 24 hours, proceed to bacterial plating and testing the household cleaning agents. C. Bacterial plating. Make available a waste container with 10% bleach. 1. Using a permanent marker, label the side of the plate, not the lid, with your name or initials and group number. 2. On the bottom of the plate, about 1 centimeter from the edge, write the number 1 and circle it. This will be used to guide you to place the filter paper square later in the experiment. 3. Repeat step 2 on the opposite of the first edge, but write the number 2 and circle it. 4. Use a sterile cotton swab to dip into the bacterial suspension. Squeeze out as much liquid as possible from the swab by pressing it on the side of the bottle. 5. Take the swab and streak the top of the nutrient agar plate going from bottom to top until the entire plate is swabbed. Then turn the plate 90 degrees and swab again. You want to create a lawn of bacterial growth. 6. Cover the plate and let the agar dry at room temperature for 5 minutes. Before discarding swabs used to inoculate the bacterial cultures, soak them in 10% bleach for 20 minutes, then discard in regular trash. Reminder: be sure to wash your hands with antibacterial soap upon completion of this activity. D. Testing household cleaners. 1. Cut filter paper strips into equal squares. You will need 1 square per cleaner to be tested. 2. Each student or group should choose 2 cleaners to test. Pour a small amount into a small disposable cup. For each one, soak a filter paper square in the soap or cleaning solution for 30 seconds. 3. Use the tweezers to pick up the filter paper square and place it on the inoculated nutrient agar in the area above circle number 1. Record on the table what cleaner was used for sample number 1. 4. Repeat step 3 with the other cleaner. Record the cleaner used for sample number 2. 5. When done, place the lid on the plate and tape it with Scotch tape. 6. Turn the plate upside down. Place the inverted plate in a safe place and incubate or store it at room temperature for 24 hours. 7. Wash your hands with soap and water after this exercise. E. Positive and negative controls. 1. Use tap water as a sample for the negative control. Soak a filter paper square in tap water for 30 seconds and then place the square on the agar above the number 1 circle. 2. Use the sample of the effective disinfectant provided for the positive control. Repeat as in step 1, but place the square on the number 2 circle. F. Record your results. 1. Observe the plates after 24 hours and record your observations on the table provided. 2. Incubate the plates for another 24 hours and record your observations in the 48 hours column. Incubate another 24 hours if necessary. G. Expected results. If the cleanser is effective in killing bacteria, then there will be a zone of inhibition around the disc. That is, there will not be any bacterial growth around the square. If there is bacterial growth around the disc, the cleanser is not effective in killing bacteria. How effective are the cleaning products in your home? The experiments you have performed answer this question. It should be obvious from your experiments that many cleaning products are not very effective. This basic procedure is also used to select the best antibiotic to treat bacteria identified as the causative agent for illness.